Today on Shay's Microbiology, we are kind of closing a chapter. We are wrapping up systemic bacteriology and at the same time slowly preparing ourselves to move into virology. But before we jump there, we really need to understand one last and honestly very important group of bacteria, and that's gram-negative rods and gram-negative diplococci. Because if we are being honest, many of the infections we see in real life, UTIs, diarrhea, pneumonia, meningitis, sepsis, they come from this group. So let's take it slowly, reason through it and finish bacteriology properly. What gram-negative actually means. So first, what do we mean when we say gram-negative? On gram staining, these bacteria appear pink or red, right? And that color change isn't random, it's structural. Gram-negative bacteria have a thin peptidoglycan layer, an outer membrane, and something called lipopolysaccharide. Now, this outer membrane, it's a big deal. It makes these bacteria harder to kill, limits antibiotic entry, and contribute to toxicity. So whenever you hear gram-negative, your brain should already be thinking more complex, more resistant, more clinically serious. Now, once we know they are gram-negative, the next thing we look at is shape. Some appear as rods, elongated cells, others appear as diplococci, round bacteria sitting in pairs, and this simple observation already narrows things down a lot. Let's start with gram-negative rods because this is where most people feel overwhelmed. But instead of memorizing names, let's group them logically. Enterobacterias, the gut group. So first, we have the enterobacterias, organisms that normally live in the gut. Examples include Escherichia coli, Klebsiella, Salmonella, Shigella, Proteus. In the lab, we usually ask, can it ferment lactose? Does it produce gas? Does it produce hydrogen sulfide? That's why we use tests like McConkey-Agar, TSI, indole, citrate, urease. Each one answering one small question. Clinically, these organisms are major causes of UTIs, sepsis, and diarrheal diseases. Now, another group, and honestly a scary one, is the non-fermenting gram-negative rods. These don't ferment sugars easily. Examples include Pseudomonas aeruginosa and Acinetobacter. You hear about these a lot in ICUs, ventilated patients, burns unit, and these organisms are often multi-drug resistant. So whenever you hear non-fermented, think hospital-acquired, resistant, difficult to treat. Some gram-negative rods don't follow the usual rules like Vibrio cholerae, curved, Campylobacter, curved and microaerophilic, Helicobacter pylori, spiral-shaped. These organisms often need special conditions to grow, and clinically they are linked to cholera, diarrhea, gastritis, peptic ulcer. Again, shape plus metabolism guides in identification. Now let's move to a smaller but very serious group. Gram-negative diplococci usually appear kidney-shaped, often inside white blood cells. The two main ones are Neisseria gonorrhea and Neisseria meningitidis. In the lab, we identify them using oxidase testing and sugar fermentation patterns. And these organisms remind us that, honestly, arrangement and biochemistry matter just as much as the stain. So if you pause for a second and zoom out, identification follows a pattern: gram stain, shape, arrangement, biochemical behavior. It's not guesswork, it's not magic, it's a logical decision tree. Once you get that mindset, bacteriology stops being stressful. At this point, we have covered gram-positive cocci, gram-positive rods, gram-negative diplococci. That completes classical systemic bacteriology. So today, we close systemic bacteriology from gram-positive organisms all the way to gram-negative rods and diplococci. And next, we move into virology, where biology behaves very differently.